Well, thanks for this kind of nice introduction. So I was asked to talk about the future developments in theoretical neuroscience. Of course, there are many types of theoretical neuroscience. So of course, my perspective is a little bit obviously biased about my background, which is in physics. So I thought I should start with this Norwegian stamp. This is actually the guy on this stamp is Niels Albert. He's a quite famous mathematician. We talk about abelian groups and even non-abelian groups. But you can also see that he died quite young. He was only 27 years old. So he died on this farm on the way back up in southern Norway on the way back from Paris. He didn't get a permanent job until 3 days after he died, actually. Then he got a professor job in Berlin. But the reason I'm bringing up now is that I read his biography. And then when he was on his deathbed on this farm, he was so upset about the doctors at the time. How he died of tuberculosis. He thought that was completely ridiculous, that we should die of this very simple disease, that the doctors couldn't figure this thing out, right? And now, at least in the Western world, we don't die of tuberculosis anymore. But if we go to the doctor and we come back with a brain disorder, say like dementia or a serious psychiatric disorder like schizophrenia, there's nothing you can do. So in some sense, we are not, or the doctors don't have much to offer. So we are not doing too well, I would say, in neuroscience. So and there, of course, there are other people also are pointed this out. This was something I read a few years ago in Wired. They had like this story of why science is failing us when it comes to development of new drugs, and actually brain drugs was mentioned in particular. And they actually said that most recently 2 giant firms, AstraZeneca, GlaxoSmithKline, this is like big pharmaceutical companies, announced that they were scaling back research into the brain. The organ is too complicated, too full of networks we don't comprehend. So not only don't we have a good treatment for these diseases, we don't even know. We don't even know what they what their origins are, and also that the big pharmaceutical companies are pulling back from research. So this is a rather serious situation. And then if you ask why aren't we doing better? So, well, then we point to all these scales. There's so many scales in the brain, all the way from like the nanometer scale of the proteins, the molecules, up through up to the cells and connections between the cell synapses, of the circuits, and all the way up to the system. And also many temporal scales. From picoseconds, that's a time scale for how molecules interact, up to, well, the millisecond time scale of this action potential propagation and the signaling between neurons, all the way up to the lifetime of an organism. So this is maybe daunting. On the other hand, we've already done it for another system. If you look at the smartphone, this is really, I think, the prime example of where mankind really has been able to bridge all these scales. I think if I could send something to like a foreign civilization telling about how far has mankind come in terms of technological development, I just send a smartphone, because there you have all this general theory of relativity is in there, certainly quantum physics, and this fantastic engineering achievement of linking all these scales from molecules all the way up to systems. So if we look at this, one way to set up these scales for the brain, all the way atom, molecule, molecular network, neuron, all the way up to the brain, we have similar scales when it comes to making a smartphone or making a computer. So we have atoms, and then we have a crystal. In that case, it's like semiconductor crystals, and then we have these tailored materials. This is what I used to work on before I got interested in neuroscience. This is what is called semiconductor heterostructure. And then you have the transistors and chip, and all the way up to the smartphone. So we already know how to bridge scales. But how do they do it? Well, the tool we used, we need a really precise language to bridge scales, and the language is mathematics. So that's how it did it. And actually, if we think of I talked to people working on the Spinnaker, the programmers working with the Spinnaker chip or Spinnaker system yesterday, and of course also there you're bridging all these scales from that you can actually compute, well, you can program with this Pine, this high-level tool, and go all the way down to manipulate the chip. That's done like 4 or 5 levels of programming, right? So this is sort of something we do. We need a precise language, whether it's like programming languages or mathematics. So that's something which has not been pursued to the same extent in, or I would say not really pursued at all, in neuroscience until now. So if we go back to like how we bridge scale in smartphones, like 100 years ago, we didn't even know why some materials really conduct electricity well, like metals, and some are insulating, like quartz or silicon, because they're just made of atoms. How can they be so extremely different? It's like many, many orders of magnitude in difference in how well they conduct electricity. And you didn't find the answer to this by doing more experiments or new fancy microscopes. You did it because you were able to come up with a theory. So this was like this quantum theory that came up to describe how atoms work, and then they realized that, well, this Schrödinger equation was not only applicable to atoms, but also molecules, and then also solid state, materials which are made up of atoms lined up in lattices. So based on that, we sort of realized why metals and insulators are so different in sort of conductivity, and then you could start making inventions based on that. And in 47, you got the transistor based on silicon, and then 58, the integrated circuit, which sort of paved the way for this mass production. So it was really and this thing was done by this tight interaction between people who do, well, in physics you say experimentalists and theoreticians. In neuroscience you say experimentalists and modelers. And this also, not only did these people who did this collaboration, not only did they sort of like work together in research groups, they were also trained together. Basically, they had the same basic training. So if you, for example, go into any kind of if you Google for like a study program in physics and engineering, you come up with this quite standardized plan, which has a combination of that you essentially have the same basic training. This is like a physics curriculum. I don't really know what university this is. Could be any university really. Has this combination of mathematics, basic physics, both experimental and theoretical. So the key thing is that the people who do the experiments and who do the modeling, they are trained together. So they learn the same language and they learn the same techniques. I think this is where we also have to go when it comes to neuroscience, that the future neuroscientists would have to be trained in one program where they learn both experimental and modeling techniques. And until then, we have to make workshops like this. No, and again, we should always have workshops, but I think it's certainly a lot of things to be done in terms of, it's just sort of striking how differently physics is done compared to how neuroscience is done. So in physics, has like this two-handed collaboration between experiments and mathematical modeling, while in neuroscience, it's essentially been a one-handed science. You do experiments, and data analysis has been sort of quite naive and not really based on like modeling of the underlying systems. So this is sort of why I'm sort of trying to advocate this kind of neuroscience with both hands, where you really get sort of look at what worked for making a smartphone in physics and engineering, and try to do the same in neuroscience. So that's sort of the bad news is that we're not doing too well in neuroscience. I would think not only in terms of not curing diseases, but also in terms of understanding how the brain works. But the good news is that we haven't really tried everything up our sleeve yet. We have like this possibility to try like different research methods to improve the situation. So now if we go into the brain, I guess we've had some talks about the brain earlier this week, and you maybe know I used to do semiconductor physics, so I learned about the brain in like when I was an adult. So I remember how confused I was about all this, like what's a synapse, what's a neuron, it's like. So I know how it is to be confused and not remember things. So I just, in case, want to give a reminder. So here is here's like a cross section through the human brain, and out there, out on the surface, out here, you have this cortex, this like 2, 3, 4 millimeter thick layer of cells, and if you sort of blow it up, then it looks something like this. This is actually a picture from a macaque monkey, but it's very similar. So we have, and all of these little dots here corresponds to cell bodies of nerve cells. So what we see is that it's like quite a similar structure if you go sideways, but it's quite different if you go in a vertical direction. So it seems like also that the kind of the brain you use to do, say, like learn language or to see or to hear or to do chess or fall in love is sort of like very much the same. It looks very much the same. It's sort of like the way it's wired, or the algorithms are different. So if you put in another kind of ink, well, this is I've taken this exposure and blown it up again, and then we can see all these stripes. But then also if you put in another type of ink, you get this kind of weird situation very nice situation, where the ink creeps into only a small handful of the cells, only a few of the cells, but those it goes into, it fills completely. So then you can see that these 3 cells are really, which you can sort of like where the ink has crept in, are really these beautiful branched structures. So, of course, if they had crept into all cells, it would have been completely black. So here we're taking some of these reconstructed cells and superimpose it on this image just to show what the brain or the cortex really looks like. So this is one cell, this red and blue thing, the red and yellow thing here. The red is where this neuron gets input from other cells, and the yellow is where it sends out to other cells, the axon. So what have people been doing? Really, what have people been doing in terms of measuring how the brain works? I would say like the most of what we learned on the brain is sort of by putting on the in the what's called in vivo brain, like the operating in the brain of living animals, is by putting sharp electrodes close to neurons, a little bit at random, but you go down here until you can sort of get to listen to the signals from these cells. And then you realize that these cells actually send out these these spikes. This action potential lasts about one millisecond. And then you can do this investigation and see what kind of things make this neuron fire these spikes of action potentials. And here's one movie that illustrates this. This is actually a rat running around in this enclosement here, and then you can see that it's this is not static, these are actually spikes, and you can see that it only spikes in certain situations, and then for this particular cell, then it's only when the head of the rat is pointing like this way. So you can actually follow the arrow and listen to the loudspeaker. You can see that actually this is the so-called head direction cell, which tells sort of signals what way the head is pointing of the rat. So this is one example of what's called a receptive field. I found this on the internet. I think it was recorded by Francesca. I don't really remember the last name, but so this is my credit goes to Francesca, wherever she is, who sort of made this very nice video and made it available. But this is just an example of what is called a receptive field. So this go all the way back to the studies of like Hubel and Wiesel in the 50s and 60s, where they put these electrodes in the back of the head here in the visual cortex, and they saw these cells that only fired spikes when you had stimuli oriented in a particular direction. So this is like what is called this direction selective cells. So, and this was a great success, and learned a lot about at least sort of like neural representation, how the neurons encode information. And then following sort of like that approach, then we have sort of like mankind has put electrodes all over the head all over the cortex, and look sort of what kind of things make these different kind of neurons fire action potentials. And actually, if you put it up here on the middle of like the top of the brain, you get these cells which are sensitive to particular like movement of fingers and all muscles, and also like sensory input. So that's like the receptive fields of these neurons. And then there's also this really famous receptive field, the Jennifer Aniston cell. So this was like this cell that only fired when like this person saw a picture of Jennifer Aniston, and it's like not one particular picture. It was actually also sufficient just to write sort of like Jennifer Aniston on like in text. So like this object cell. So we all go around and have Obama cells and Putin cells. So certain cells that only signals now if we think about or we see a picture of Obama. But just this is, of course, this is done in humans, but you never put in these sharp electrodes in humans just for research practices. This was done because, well, while they were doing recordings to prepare for epileptic surgery. So it's only like a byproduct. And then if you come from Norway, of course, our favorite receptive field are these grid cells, which were discovered in Trondheim in a Moser lab, where it turns out that you have certain cells that only fire when the rat, in this case, is in a particular position. And these were sort of like really peculiar cells in that they didn't only fire in like when the rat was in particular position, but in this like a grid of set of positions which together form this grid. So this is really, I would say, this is what we have really learned about the brain in some sense. And it's a little bit, in some sense, it's really cool. But also this thing was done 50 years ago. And we're still measuring receptive fields. That's more or less what we do in experimental neuroscience. And it's sort of, while we learn a lot, it's also limited how far we can get. But in terms of modeling, what people, of course, then have focused a lot on is modeling of spikes. Modeling of how neurons generate spikes, and also how spikes are generated in networks. And we know how to model neurons. That's one of the like few anchors of sanity. That's maybe putting it too hard, but there's like one of the things we know how to do in neuroscience. We can do this, what's called multi-compartmental modeling. So we can take this neuron and split it up into these compartments, and these compartments has to be so small that the potential, the electrical potential, which is like the key dynamic variable, doesn't vary inside the compartment. And then we can, so then we set up this equation set, which is based on Kirchhoff's current law, the same kind of law that you use when you construct the electrical circuits, just saying that currents cannot vanish, or charge cannot vanish, so current has to sum to zero when you sort of into a node in the network. So it's just keeping track of all the currents. In the computer, it's electrons. So that's why computers are so fast. Electrons are really light, so you can really switch them at gigahertz, like billions of times per second. Neurons are, there is not electron, it's ions. So it's atoms that is much more sluggish. So that's why neurons are much more sluggish than transistors. So we're talking about like 100 hertz, 200 hertz on a good day, right? So a neuron is really slow compared to like a transistor. But anyway, we can keep track of all the currents, and we get this set of differential equations, which can be used then to simulate how these currents and potentials spread across this neuron, and how these spikes, these action potentials, are generated when you get synaptic input. Actually, the first neural simulators was based on PSpice, which is this tool that is used to simulate electrical circuits, just illustrating that it's really, really the same thing. But if you do that, then you can actually do this kind of simulation. This is a simulation that Espen Hagen did when he was working with us. So this is like 5-6 electrons, and not electrons, 5-6 neurons, which are, you can see, is reconstructed, and the color code tells you about the membrane potential, which is about minus 60 millivolt at when nothing is happening, that's like the resting potential. But then you can see that they occasionally get this, they occasionally fire these action potentials, which you can see as these flashes. So this will then fire an action potential. And this gray or like whitish pencil back here, that's like the recording electrode. The point here is we know how to, this is like in our model world, we can do everything. We know how to simulate neurons. And of course, if you start then if you want to simulate many of these neurons, then you need strong computers and supercomputers and so on. That's of course had to do with the previous talk. So then, so I say that, so this is also then the goal of the one of the overall goals of the Human Brain Project is to link all these different scales by means of mathematics. And in order to solve this system of mathematical equation, you need strong computers and even make new really strong computers and new ways of doing computing. Because the good thing is that we know essentially the laws of how the nature operates at these different scales. At the molecular scale, it's really just Newton's law plus some quantum mechanics. At the more like at the synapse, inside the synapse, it's more like these chemical rate equations. And up at the neuron level, it's the cable equation and the diffusion equation and so on. So we know the basic physics, or the physics equations that we need to simulate the brain was sort of discovered in the 20th century. So now we just have to put all this different information together. So now we want to then to make these mathematical models, but I would say like biophysics-based mathematical models for how like the brain or pieces of the brain operate. But then in order to do science, we have to compare with experiments, right? We have to compare with how nature is. So you need to compare with experiments. So then if you focus on like, if you focus on the cortical circuit, a piece of the cortex, like I said, the typical way to test to test your model has been against these spikes. So you put in a sharp electrode and measure like the spikes from individual neurons, or maybe like if you have like many electrodes, maybe from a handful of neurons. If you're really good, then you can also actually measure the membrane potential, but then you have to have this sharp electrode that impales the neuron. But there are many other now measures of neural activity. You can then put in this other kind of electrode, which has many contacts, but each contact is not so sharp. So, and then instead of picking up like signal spikes from one neuron, you actually pick up either like the spikes from like many neurons surrounding the contact, or even the what's called which is contained in the high frequency part, high frequency meaning about 500 hertz or so, or the so-called local field potential, which is the low frequency part, which maybe comes from thousands or even millions of neurons. And then also we have a bunch of like a whole new set of techniques based on optical techniques, sending in light and see what comes out. So then there's all kinds of tricks you can do and add different kinds of like fluorophores and so that you can sort of get higher selectivity and so on. But that's the basic principle. And then you also have these more like systems level measures, functional MRI, which measures the oxygen consumption and blood flow in the brain, PET, which measures the essentially food consumption in different parts of the brain, and then EEG, MEG, which measures the electrical signals and magnetic signals outside the skull. But these are more like systems level measures, but you can also then, of course, this is really just reflecting what's happening in a larger part of cortex. So then we have all these measures of cortical activity. So what has been the traditional way of analyzing these data? Well, the really traditional way has been to look at spikes and then look for correlations between the spikes you measure and what the animal is doing. I like either whether you sort of, if it gets some optical stimulus or like visual stimulus or auditory stimulus, or what it's or if it's behaving like moving around in in a room, or even like pointing the heads to different directions and so on. So that's what is called these receptive fields. But now, when people are so good, they can do more experiments and actually measure several things at the same time. What did you do then? Well, typically they do this what's called, typical multimodal analysis. Multimodal means that it's more than one experimental modality. Then you look for correlations between different experiments. So you measure two things at the same time and say, well, how is the multi-unit activity related to the two-photon calcium imaging statistically? You look for correlations and all kinds of and then if you're really good at statistics and you're lucky and you work hard enough with your data, maybe you find out with something which has some kind of feature of something that has sufficiently small p value. That's like the game of statistics. It seems to get sufficiently small p values, and if it's sufficiently small, you declare victory and publish your results and then move on. But it's really unclear. I think it's very often very difficult to make progress that way. And I think that was also pointed out in this Wired article. So they say, but here's the bad news. The reliance on correlations has entered an age of diminishing returns. At least two major factors contribute to this trend. First, all of the easy causes have been found, which means that scientists are now forced to search for ever subtler correlations, mining that mountain of facts for the tiniest association. Is that a new course or just a statistical mistake? And I think often it's things which are published, turns out this is like a big problem, which is acknowledged by many, that research is not like half of the system, or like life science research is not reproducible. The things which are published, when people try to do it again, then you don't get the same answer. And that's, of course, that's like almost like a death sentence for science if the things you do are not reproducible. So even like the big pharmaceutical companies are now saying we can't trust basic science anymore. So they had like, we're sort of now saying that this is such a flawed process that that we cannot really rely on these data to start like testing on patients, or testing on mice and testing on patients, because they had some really, really costly mistakes, some of these pharmaceutical companies, where they are like trusted ideas from well, at least they started based on existing basic science and tried to develop drugs, and which have then turned out to not work at the end. And this costs literally billions or like hundreds of millions of dollars to test out. So we need something else, I think. And so if you look at these measures of cortical activity, if you look a little bit closer on them, you can see that actually many of these measures are just different aspects of the same neural activity. So if you have one neuron here, then the spike you measure, if you have a sharp electrode placed here, it's actually a weighted sum of the transmembrane currents in the soma region. That actually follows from the basic biophysics of this measurement. While the LFP, the local field potential, and the contribution to EEG and MEG is the weighted sum of the transmembrane currents all over the neuron, not only in the soma region. While the voltage-sensitive dye, that's one of these optical techniques where you put in some kind of ink, and then you send in light, and the signal you get out is actually proportional to, it's like a weighted sum of the membrane potentials, not the transmembrane currents, but the membrane potential close to the cortical surface. But the key thing here is that the same, if you know the neural activity, you can compute all these measures simultaneously. You don't have to just rely on statistical correlations. So we need to work out the mathematical connections between neuron dynamics and the different experimental modalities. It's called like the measurement physics. And I think this is something which has been like in computational neuroscience, this has been far too little emphasis on this. Like for one thing, there's very few computational neuroscientists, meaning people with like training in like physics or engineering or computer science or maybe applied math, going in making like physics type models or even models at all. So it's been difficult to form a community, so that the community is actually focused on, I think, often support very specific, they develop very specific models for specific data, but also they've been typically focused on getting the right spike dynamics. So they're really testing their model against measured spikes. And I think that is sort of too limited. We have now to go beyond that and develop these links to these other measurement, proper links. And for example, if you look at, I mean, a couple of years ago, this Higgs boson was discovered in CERN. And if you look at the people who work at CERN, right, there were like 20, 30 people who actually worked on like the theory of why there should be a Higgs boson. And this was just like a support thing. This was actually the predicted Higgs boson, why quantum theory predicts a Higgs boson based on the standard theory of that. Just was something was developed in the 60s by Higgs, for example, and others. So everybody else just worked on the detector physics, like the measurement physics. So we need also, like in computation, in neuroscience, we need a much more emphasis on doing this measurements physics properly. And I think that's one of the things, like talking about developments of theoretical neuroscience, this is one of the things that we have to cover. So the perspective, I think, for models and model testing, really, that is that you should, in order to make sense and make sense of the brain, you need to start with a specific model, or like a candidate model, like a test, like a preliminary model for how like your piece of brain works. And then they should be formulated in mathematics, because they are the only language is precise enough, really, to test the model specifically. This goes back to Galilei, saying that the laws of nature are written in mathematics. That certainly also applies to the brain. But then the model should be compared with all measurement modalities. So say if you have if this sort of is your model for how a particular piece of like a neural circuit, maybe with like some 10,000, 100,000 nerve cells, how it behaves, then it should not only be compared with spikes. It needs to be, you need to develop the link between all this, between the activity in the circuit and all these kind of measurements. We have done a lot of work on making the link between like activities in circuit and this local field potential, because this is like a very common measure and there's a lot of data existing, but this should be done for all these measurement modalities. And this is what I would like. I mean, I like to ski, like cross-country skiing in Norway, and now, I mean, it would be wonderful if I could sort of like, because this is where you like to be creative, right? This is where maybe the brain does this. What is the role of this channel? What is the role? So I would like to be able to think about what the model looks like, or what the model should be in order to do the information processing and so on, and then be able to test this easily on a computer. Maybe I could even have like a laptop computer in the Norwegian mountains on my hut. So I come back after some ski trip and I think, well, I wonder what the role of this L-type calcium channel is, and then I can just log into my sort of like my some kind of simulator on some super duper computer in maybe Jülich or elsewhere, and not only simulate the spikes, but given this model, be able to make predictions for all these things. And this is something which can be done sometimes. This link here can be done in some sense once and for all. If you do it properly, this should sort of be something which is like a tool which can be there for everybody to use. So we also then need these neuroinformatics tools to make this as simple as possible. We have made this tool called LFPy, which is how to go from like neural simulations on the type you do with like this blue brain type of multi-compartmental models, and to link this to the LFPs. So now I'm going to focus more on this measurement of these electrical potentials, and we'll be more specific on the local field potential. So this electrical potential, this is the measurement of these electrical potentials are among the oldest and conceptually simplest measurements of neural activity. It goes back to this Richard Caton guy, I think, 1875, so he's almost 150 years old, who measured these electrical potentials from surfaces of animal brains. So what are these measurements? Well, you take this, look into a piece of cortex, for example, and you put down a recording electrode and measure the potential difference between the tip here and a reference electrode far away. And then you measure this, then you measure potential, which is about a millivolt, a little bit less than a millivolt. So it's much smaller than the membrane potential, because this potential is much more like it is. It's like the life is less resistive in the extracellular medium. So, and the reason we got interested in this is that this is I just came from California, so I'm still a little bit jet lagged, where I met again with Anna DeVore and Anders Dale, our two collaborators now at UC San Diego. And Anna, some 10 years ago, she did these measurements where she put this laminar electrodes, it's like a multi-electrode in many contacts, across what's called a barrel cortex. So it's like a piece of cortex allocated to dealing with whisker processing. So the rat has 30 whiskers on each side of the snout, side of the face. And so they are used for navigation and so on. They can also whisk it themselves. So it's a little bit more than our mustaches. But it turns out that each of these whiskers have a dedicated piece of cortex to them. So it's really this fantastic system. So you can sort of look at this chessboard arrangement of whiskers on the snout, and you can refine it inside the cortex. So meaning that if you flick the C1 whisker, then you get a big response in the C1 piece of cortex, which is called like this. It's called a barrel cortex, because if you look at this from the side, it actually looks a little bit like a barrel. So then what happens if you flick a whisker? So here is like the measurement of the electrical signal from the top of the cortex to the bottom of the cortex. So this is like 2 millimeters. So about 10-15 milliseconds after you flick the whisker, you see this volley of activity, this electrical potential, about like a little bit like a half a millivolt or up to a millivolt or so, all across the cortex. And if you look at the high frequency part, most of the power is in the low frequency part. So the high frequency part, then you get so then you get this quite different pattern, but this reflects more the firing of action potentials of neurons in the vicinity of this electrode contact, while this reflects more the inputs to the neurons in the vicinity of the electrode contacts. So how can we use this? How can we make progress with this kind of data? Well, I think what you need to do is you need to formulate then a network model, and then translate your network model, meaning that like this is neuron here, neuron there, and they're connecting this and this, and be able to connect your network model then to things you can, like these things that you can measure. And the really good thing about these electrical measurements is that we know their physical origin. We know that the source of the extracellular potential are these transmembrane currents, meaning that if you measure the extracellular potential here, the potential difference between this tip and the reference electrode far away, and if the only thing that happens in your piece of cortex is this orange neuron receiving a synaptic input up here, then we know that current, an excitatory synaptic current, will give current going into the neuron. So from the point of view of this extracellular electrode, it will mean that there's like a current, it's called a current sink. And it follows from the basic multi-compartmental modeling scheme, cable equation, that this current has to return somewhere. So then if for now simplicity, assume that it all returns through the soma, then you have a current going in here and a current going out there. And then if you know the distances between this current sink and this point here, current source, and the electrode recording tip, then we can actually compute the potential. So this is called a forward solution because it links the currents, the neural activity, to what you should measure. And the good thing is that this is a linear theory. So even though processing of spikes, and that's like a non-linear and very complicated, but the measurement physics linking neural activity to what you can measure, that's linear because electrostatics is a linear theory. The only parameter that goes into this is this extracellular connectivity, which tells how well this neural tissue actually conducts electricity, and that is something which can be measured separately. And since it's linear, this directly generalizes to the case where it's not only two compartments in the neuron that has transmembrane currents, but when you have many compartments which all have transmembrane currents, it's just the same thing. You just have to sum up all the contributions, like weighted with like the transmembrane currents weighted by the distance to the electrode contact. So meaning that if we already do this multi-compartmental modeling, what we need to keep track of are all these currents going through the cell membrane, like these things here. And if you do that and use this forward modeling formula, we can then compute not only the membrane potential, which is typically what you sort of, if you want to predict spikes, this is what you would measure on the outside. This is a system where like 5 neurons, and it's a little bit boring because it's like green most of the time, which is 0. But occasionally you see that there are these flashes down here. You see there was a big flash there, which is this action potential. So that tells you if you want to measure spikes from this neuron here, you better put your electrode here, because then you get a big signal. If you put it out here, you get a much smaller signal. So the key thing here is that now we sort of have a system where you can both compute the intercellular potential and the spikes, maybe like which you think is which is what is important for the information processing, but also the extracellular potential, which is important for measuring, for the measurement part. So we have explored this sort of quite a bit, how they sort of, that was the spikes mainly, but if you want to look at the LFP, the local field potential, the low frequency part, and then we can, so for example, we have an example here where we inject the synaptic input here, and then what we see is that if this is the synaptic input current, then you get an extracellular potential that looks like, has the same shape, and it has quite sharp and a large amplitude if you have the electrode close to the soma. No, close to the soma, close to the synaptic input. So this sort of is sort of quite simple to interpret. However, if you go down here, even outside the soma, you get big signals, even though the synapse was up here. So if you measure like this thing down here, it doesn't mean that anything close to this, I mean, there was like a synaptic input close to the soma. It might as well be that you get the synaptic input high up here. And actually, if you look at, and you see another thing also, this thing is not only lower amplitude, it's inverted, but also it's not as sharp. You lose some high frequencies on the way, which has to do with the cable properties of the capacity properties of the cable or the neuronal cable dendrite. So this peak is actually 5 milliseconds after this peak. And in the past, this has led people to conclude that this must be a separate process. This is some kind of, this is one process, while this is like in a input current with the opposite sign at the soma following 5 milliseconds afterwards, sort of implying that there's some kind of following inhibition after excitation and so on. And this kind of, which is, well, as we show here, this is not the case. In this case, this is not the case at all. This is just sort of, they haven't done the measurement physics properly, so they infer this, they get these incorrect inferrals. And this, this is also very, like, has been very important when it, or this kind of mistake has been very prominent when it comes to interpretation of the so-called power spectra. So this, one way to analyze data is to sort of take the signal, and you take the Fourier spectra, look at frequency content, and you look at how the signal power varies with frequency. And then you make inferrals on what the system is doing based on that. So typically when people have analyzed this in the past, they have sort of assumed that there's that the only frequency dependence that you see in the, for example, the EEG has to do with the frequency, different frequencies in the firing rates. So they have made these firing rate models, or like models for the spike firing rate and so on, and compared it directly with EEG. But look here, here you can see that if you drive this is like a neuron that you drive with like a 1 Hertz oscillatory current input. So you see, you get this nice current dipole. And what you pick up at the EEG, with EEG or MEG are like the summed contribution from millions of such dipoles. But then if you do it at 100 hertz, you would think maybe you get the same thing just 100 times faster. But you don't. I'm just able to get this in, as you can see here. So you get a very different pattern. You get a much smaller dipole. It's actually more like what's called a quadrupole. So this thing, just because of so even if this input has the same amplitude, you get a much smaller dipole and a much smaller contribution to the EEG signal, just because of this, like the cable properties of the neuron. So that means that when people have, for example, made theories for these EEG power spectra in the past, where they take this log, they plot the power, like the squared amplitude as a function of frequency, and you get these like power laws here, and they make all kinds of fancy theories. If you don't take this biophysical, which people haven't, if they don't take this biophysical effect into account, it has to be wrong. So it just illustrates that it doesn't help if you have a good theory for what the network does in terms of generating spikes. If you want to compare with experiments, you need to have this other theory linking the activity in networks and neurons to the things you can measure. And that has to be done for each measurement separately. One thing we have done is this, with this biophysical modeling approach, is to look at this question: how local is the local field potential? Meaning that it's called the local field potential, and the reason it was called a local field potential was that when you before they put the neuron inside the brain like this, they measured EEG. So there it was less local, meaning that there were more neural sources contributing, because it was like the detector, the microphone, was further away from the crowd. But if you put like the electrode inside the cortex, it gets more local, meaning that you pick up signals, you pick up this contribution from transmemory currents in the neurons surrounding the electrode tip. But how big is this basin? So this has sort of not been really been known, and the experimental, like the different reports, there's been experimental reports on this has sort of varied widely, all the way from like 0.2, 0.3 millimeters to up to a centimeter. And this is very unsatisfactory, given that this is such an important we spend millions and millions of dollars doing these kind of measurements, doing these kind of recordings, and we don't even know how local the signal is. So we did an investigation of this, and by using our biophysical modeling scheme, and asked the question, how big is this spatial reach? And then first we looked at, what are the things that determines this spatial reach? Well, one thing, if you have this electrode here surrounded by a bunch of neurons, then we know that all of these neurons are going to contribute, but the neurons which are closer will contribute more, because it's like the signal decays away from the neuron. The other thing is also, however, is that if you look from the geometry, if you ask the question, there are more neurons at distance, if you divide this region into rings, there are more neurons in the rings with a large R than a small r. That just follows from the geometry. So that's like an opposing effect. But the third thing, which turns out to be actually most important, is how synchronized are the LFP sources. So you can get something analogous to this, like interference effects from electromagnetism or wave mechanics. and this turned out to be the key to understanding why people have measured so different values for this spatial reach. So what we found by doing a thorough study with large-scale simulations of populations of neurons and so on, that if the neurons around the electrode are uncorrelated, meaning that they just get so they just get random input essentially, then the spatial reach, it's a very local thing. It's only like 0.2 millimeter. However, if these neurons are like a choir singing coordinated, then it can be much larger, and it's really the size of the choir that matters. If the choir is 1 millimeter and they talk completely 1 millimeter sized population around the neuron, if it's 1 millimeter, then actually you pick up, then the spatial reach is is 1 millimeter. So it's really this spatial reach is set by the spatial correlations. Okay, so now this is more like principal issues about like what LFP is. But what we really want to do is, I want to be able to make a network model and be able to compute LFP so that I can use the LFP to test my theory. And this is, and often when people, I don't know if you had any talks about the nest, with nest? Has there been some? Yeah. So many, actually this is, if people want to make, when people now, and this is also an important part of the Human Brain Project, is to make these nest models like point neuron networks of like pieces of the brain, pieces of cortex, say. But these point neurons, there are many good reasons to use point neurons, I think, but they don't generate extracellular potentials, because in order to have a neuron that generate extracellular potential, you have to at least have two compartments. So a point neuron does not generate extracellular potentials. But nevertheless, I would like to be able to make a model with point neurons and then be able to predict LFPs. And then you have to do some tricks. So this is what we call this trick we have developed, what's called there's a hybrid LFP scheme, where we look at this, start with this nest-based point neuron network. So this is actually something called the Potjans-Diesmann model. I don't know if somebody had mentioned it here. It's one of the few nest models which are available for everyone. It's a piece of like a 1 square millimeter patch of I think, visual cortex with about 80,000 neurons. So we use that to, like in this case, to generate the spikes. And then, given these spikes, we combine these spikes, which is the output from the nest simulations, with information about what these neurons really look like, not the point neuron, but the real reconstructed neurons. And then we use these spikes and set up this other network model, also with 80,000 neurons, but then 80,000 multi-compartmental neurons, which we then can use to compute the LFP. So now I think I even have a movie prepared. Let's see if I get that to work. Okay, so what this shows is this is then just this simulation of, so this is then a simulation of a network, 80,000 neurons, and we show the spikes of just like some of them here, just to give an illustration. So you see like this is quite sort of like a random between this thalamic activation here. It's sort of quite like a random, like it is called asynchronous irregular state. And then for this, then we also hear that, well, this network at time 900 gets this input from the thalamus. So that starts, it makes this volley of activity in layer 4, which spreads out all over the cortex. But the key thing here is that we now, with this scheme, can also compute the LFP. So these curves here are the LFP, while the colored thing here is what's called the current source density, which tells about the currents that enter or leave the neurons. So this is just to illustrate that now we actually, with this kind of tool, we can now, which is a tool that doesn't require super duper computers, it requires strong computers, but it's actually a tool where you can actually do science. So you can play around with a network here and see what you get out for the LFP. So then what you can do then is, sort of, if you have different ideas about that, like different kind of networks or network states, then you can compute both the spiking activity and also the LFP and the power spectra and what have you, so that you can actually do science with it and then compare experiments with the LFP generated by a candidate model. So instead of just looking at experimental data and doing some kind of statistical things, you can actually go back and make a candidate network model, predict LFPs, and sort of and then compare with experiments. So I think this is really a key step, very important step to move forward. That we store our preliminary idea about how your piece of cortex work in terms of neurons and neural networks. But then from that, compute the things that you can measure. Like in this case, the LFP, but you can also do like the voltage-sensitive dye, all kinds of optical methods, and so on. So, of course, then the ultimate goal then is, if you have all this, or one one important goal, if you have all these scales, is to be able to go all the way from like setting up a model with neurons, and maybe also starting with the lower than neurons, like with a level below, and go all the way and be able to predict things that you can measure in humans, like non-invasively, like this like a virtual EEG generated by a model. This is just like just to illustrate what the goal is. The underlying model was very naive. But the point is, this is what the model predicts the EEG to be on this person. And this is what the same model predicts the functional MRI to be. And this is actually what the same model predicts the what's called the voltage-sensitive dye imaging image, the image to be. But that's not possible to do non-invasively at the moment. So, I don't know if anybody talked about this Erdapfel earlier in this course. I think this like a human brain project has gotten, quite a bit of attention and critique. And I think the main misconception really has been that, at least the way I think about it is, is that, this is actually this Erdapfel, which was made by this guy. This was made it in 1493. This is like the first globe attempt of a model of, there was like a model of how what the world looks like. And this was made just after Columbus came back from like what he thought was India, which we now know is America. So this guy made up this model for the globe, where you go directly from Europe to India or to China, right? So this was sort of like what is good about this model is precise. It's testable, right? You can take this globe and say, well, then I should go to this, and I should end up at, I don't know what this is, maybe Taiwan or something. Anyway, so this was testable and easy to prove wrong, which they soon did. But it just illustrates how important it is to have a precise model. When you have a precise model, even if it's wrong, it's much easier to make progress, because then you can make improvements on it and build on it. And that is really the key thing about like the Human Brain Project, and as I see it, at least the part that has to do with the model building, is not to find the model of the brain. It's just that if somebody has an idea what's the importance of this ion channel, what's the importance of this neuron, there's a way to test it. Well, what if you assume this? Then you get out the prediction in real time, which you can sort of learn from and compare with experiments. So I think it's, if you draw the analogy to Newton had like Newton did really many cool things, but what he's most famous for is that this law of gravitation, right, where two masses attract each other. And this, of course, then turn out to be, gave you the planetary orbits and so on, turn out to be correct. But given, if you had this, in order to sort of test this model, that the two masses attract each other, he needed to invent a new mathematics. Because if you want to make sure that your theory is correct, or to prove or test your theory, then you have to make predictions. And then you can, it's not trivial to go from like his law of mass attraction to the planetary orbits. You need a precise language. So he needed to invent this like a calculus. And I think what we need now is that now, in order to test our predictions, calculus is not good enough. We need these big simulators. And then people can have all kinds of ideas about what like the law of attraction is, and what are the laws setting up the brain. Then one thing, I guess you can all see the elephant in the room. One thing which is a big challenge, which we have to deal with, is when I used to do semiconductor simulation, I solved the Schrödinger equation for like semiconductors, which was quite hard. But I knew the parameters. It was like the Planck's constant, the electron mass, and so on, like 3 or 4 parameters, which are known to like 10 digits. In the brain, it's not like that. So a typical project may look like this. This is actually, since I'm sort of giving it a bad rap here, it's actually one of our projects, where we looked at this interneuron. This is this one neuron in the LGN, which is a station between retina and the visual cortex. So we are interested in that. So we had this, we wanted to make a model for that, right? And then we did this, had these collaborators, experimentalists, who did this, like this patch electrodes, which sent it this current in the soma here, and he got out some experimental spikes, depending on what kind of holding current and stimulation and so on. And then Geir Hollis in our group did what most people do. He sort of looked at what is known about the channels here, and then he fitted the values, the density of these channels, to get something that looked quite similar. And then we declared victory and published and moved on. So at least now we have these models. But what does it really mean when you have all these different fitted parameters? Right? What does it really mean? I mean, is it just curve fitting, or does it tell something about the system? And it turns out that many of these densities, these ion channel densities, which is typically what you fit in these models, they not only that they vary between neurons, they vary over time in the same neuron. So it probably is that what we're getting wrong here is that we need to go one level below and see how these ion channel densities are regulated. So you get learning rules or plasticity rules for these ion channel densities, and they will have fewer parameters. And so I think it's, but anyway, so this is a big challenge we have to deal with, how to deal with all these unknown parameters that vary over time. But I don't think it's hopeless. We just have to, I think we have to using, especially when it comes to neuron modeling, we're using maybe the wrong glasses. We have to go one level below and look at how the regulation of these ion channels. The final thing I want to talk about is there's like this other big development in life science these days, and that's sort of the genome. And the human genome was mapped out like maybe like 10, 15 years ago. Then we sort of got this rough map of the genes, how how really the 23 different chromosomes, how the things are, how they're organized, and what the different parts encode for, and so on. And not only was it sort of like, it was like a fantastic success, I think, of like fantastic achievement to be able to map out these 3 billion base pairs in the human genome, but what is even more amazing, I think, is now you can do it so cheaply that I had my genome checked like by 23andMe, this company, and found lots of new relatives, actually, DNA relatives. Turned out to be a lot of Finnish in me. I think it's because my mother came from the north. It's a lot of Finns up there. But anyway, so it's so cheap that you can sort of now do all these genome studies for many big groups. And then, of course, what you hope when you got this big map and all these cheap tools or cheap ways to measure the DNA of different people, you can start comparing groups. So say if you have like in this, what we looked into, schizophrenics, you can sort of look at what are different between the genes of schizophrenics compared to the genes of healthy controls. And it turns out that you need a lot of patience and to do this, to get the statistic right. But last year, it was we had enough. They had enough schizophrenics, essentially 40,000 schizophrenics collected or organized by this large schizophrenia consortium. So these were like patients from all over the world. And what we could have hoped is that there was only like a couple of places on the genome which was different, right, with schizophrenics or these big groups of schizophrenics and healthy, because then you could fix them manually, at least sort of like test them out and sort of maybe understand the disease better. However, that was not the case. It's very inheritable, so we know that it's like but it wasn't like one or two places on the genome. It turned out there was like 108 schizophrenia, like 108 positions on the genome, which turned out to be statistically that's significantly different between schizophrenics and controls. So what you do then, it's a little bit discouraging if you want to do animal experiments and try to sort of to block each of these 108, and of course it's combination. Each of them doesn't give schizophrenia, it's like some kind of combination of them that does it. So that's a little bit discouraging, but there actually modeling comes to help us. Because if you look at what, you can go into then this map and then look what are these 108 things coding for? And many of them code for like the excitability of nerve cells, actually the ion channels. So, and then we can actually, so then what we have done is sort of to, this is just like an illustration of a actually potassium ion channel. So what we did, we sort of looked at this. Took this for like, well, looked at these different like candidate, these genes that were like genes that was identified by these GWAS studies called genome-wide association studies, and looked at what they did in a model. And so this is just something that comes out, and well, has a paper that just came out. So then this Tuomo, then we went into the literature and looked at like what is known about the genetic variants of these ion channels, which was identified, and how how the currents of them are slightly different than the control ion channels, normal channels. And then we looked at the consequence of that in this. How does that change the properties of this cell? So in some sense, we try to make a little bit sort of like a schizophrenic test cell. And then what we found, actually, that some of them, that there is this, of course, this schizophrenia has a lot of, I'm certainly not an expert on this, and it has many like aspects to it, like many phenotypic aspects. But the one thing that many of them have is that it's called this pre-pulse or lack of pre-pulse inhibition. Because when we are sort of like, when you get this like sudden stimulus, you startle, but the second time you get it, you startle less, you sort of adapt. But many of the schizophrenics don't. So it's a little bit like, and so that we found actually the analogy to that on the single neuron level, that we found is that the control neurons were sort of like got a smaller response to a second input, while like the schizophrenic neurons got less adaptation. And so because this is actually something you can see in the EEG. So of course, this is just a prototype study, but at least I think it's a way to finally link genes to this computation, to theoretical neuroscience at this higher level. Because this is sort of something which has been lacking, not because people have thought genes are not important. That's sort of impossible to believe, but because it's been no natural way to link it. So I think we should so what we need to do, I think, is we can look at this table of ion channels. If you think of like, this is like the table of elements, like 100 elements that is together sets up the world. And then in some sense so then if you're an engineer, then it's just about putting these atoms in the right order. And then you have, this is how you invent, right? Clever ways of putting these things together. And same for a neuron. This is like a table of the ion channels. This is, I think it's like voltage activated. This is just maybe both potassium and calcium, and maybe I don't know if sodium is there. But anyway, so you have this table of ion channels. The way to think about it, maybe this is how the neurons. This is like the repertoire of neurons are set by choosing these ion channels, and then you have these genetic variants, which is maybe like a little bit analogous to these isotopes. So different ion channels, which comes in like different flavors, which is like a little bit to this different nuclei in different isotopes of atoms. So anyway, so I'll just like end with these slides where I think there's now finally a way to, I think, link genes to the overall program of computational neuroscience. So if I want to summarize like the key things, I think, which will be the development of, future developments in theoretical neuroscience. I think one is this, that you need to develop these multi-scale models, models at different scales, and they also needs to be linked. Like the best analogy is maybe this, my physics hero, Boltzmann. He was the one who found out how this laws of thermodynamics, pressure times volume is like the kBT or something, it's like the ideal gas law, how that is linked to the Newtonian mechanics of the underlying molecule. So he bridged the scale from like a Newtonian description of each molecule in a gas to this thermodynamic description. Of course, it's the same thing, it's just different glasses you put on, but he showed how it was done. So he was able to link these scales with mathematics, and this is also not something we need to systematically do, how to link these different scales. And that is also certainly part of the Human Brain Project. And then we also need to make a much larger effort on linking your models to things you can measure, do the detector physics properly. And then we have to deal with sort of these unknown parameters, meaning how they face that problem, and not just sort of like cover it, put it under the carpet. But I think there's ways to do that. And then we also have to really develop sort of what we call like modeling hygiene. If you actually, I've tried several times to have like master students start on previous models and then build on that in their master work. And I've tried 3 times, and it has never worked, meaning that either there's something wrong in the description or the parameters, something is wrong. And even sometimes I remember that just after I entered physics, I entered neuroscience from physics. It was like actually my first master student. So there was this model that we wanted to reproduce, and it was just not possible to reproduce. Something was wrong in the paper. And then we wrote to this author, and he got annoyed and said, well, if you want to do modeling, you have to make your own model. So this thing that you should build on other people's models, that was something that was even insulting to him. So that is sort of like completely ridiculous, right? If we can't build on each other's work, that's sort of like a hopeless approach in neuroscience. So but I think things are changing, really. I mean, it's a fantastically, rapidly changing field. So I think it's a wonderful opportunity if any of you, I mean, for those of you who want to go into this field, now is really the right time, and a lot of excitement around it. So thank you.